Good morning, viewers. You're welcome to our lecture series on biological techniques. Today's lecture will be on the preservation of small insects. We have looked at the preservation of larger insects earlier. So we want to look at how these small insects can be preserved, mountain small insects. These small insects have a unique way of preservation. Insects that are too small for mounting directly on standard pins are double mounted on card points, card platforms, minuting pins, or in a gelatin capsule. They can also be glued to a pin. Now I want to look at the first method for the preservation of small insects. That's the use of card points. Now, what are card points and how are they used to preserve these insects and arachnids? Fragile or tiny insects such as mosquitoes, small bugs, wasps, and flies, which are too small for a pin, will need to be glued to the point of a small triangular paper punched out, which has already been stuck through with a pin. These triangular papers are called card points. Okay, so the paper is cut into a triangular shape and the insect is pinned, is glued to the triangular tip, the tip of the triangle, which is already pinned through with an entomological pin. And that's where the insect can be preserved as long as that of the one that's pinned with the entomological pin directly. This mounting technique is explained below. The first step is, under a dissecting microscope, position the insect with the ventral side up on the edge of a small box. Then the second step is carefully spread the legs using forceps or fine needles so that the mouth parts and genitalia are not obscured. Then the third step is that you should place a small drop of glue on the tip of a card point mounted on an insect pin. Then after that, you now press the drop of glue on the card point tip against the right side of the insect thorax. So you now press the drop of glue on the card point tip against the right side of the insect's thorax, on the thorax of the minute insect. Now this is exactly how the card point is being done. The paper is first cut into a triangular shape, and then the entomological pin is used to pin through the paper, and then a glue is then placed at the tip, and then the thorax of the insect is glued to the tip of the paper. And this method is called the card point. It is called the card point. The next method we want to look at is called the card platform, or the card platforms. The card platforms are also used for the preservation of minute insects. Small insects, particularly certain beetles and parasitic wasps, but not bugs or moths, are suitable for mounting on card platforms measuring 5 × 10 millimeters. This mounting method is explained below. The procedure for preserving insects and the card platforms. Under a dissecting microscope, pin the card platform to the surface of a pinning block. Now the card platform is pinned to the surface of a pinning block. Then place the insect upside down in front of the card and spread the legs and antennae. Then the third stage in this procedure is when the glue starts to dry, arrange the legs and antennae neatly around the body. Then the fifth step is position the platform with the mounted insect on the pin using a pinning block. Now here is a picture of how the insect is being pinned. Okay, first the paper is pinned through, and then a glue is placed on the paper, and then the insect is then glued to the card platform, and this is what we call the card platform. This technique is for mounting very small insects. Now I want to look at the next method for preserving minute insects. It is called the minuting pins. The method is called minuting pins. These are used for very small moths and other small insects such as flies and bugs. Example, jumping plant lice. Stainless steel minuting pins are small. They are usually 10 to 15 millimeters long without heads. Okay, so the minuting pins are actually different from the entomological pins. They are without head, and they are the ones that are used directly on the insect. Now the procedure include the following. First, under a dissecting microscope, pin the specimen through the thorax onto a mounting board. Okay, under a dissecting microscope, pin the specimen through the thorax onto a mounting board. The insect is pinned through the thorax with a minuting pin. Then the second stage, arrange the legs and antenna with a fine forceps and brace them with minuting pins. Secure the body of small moths by bracing the abdomen on either side with the minuting pins. The next stage in this procedure is position and brace the wings and antenna with minuting pins. Remove the bracing pins when the specimen is dry. Then push a large insect pin, that size number 3, through the end of the strip of cork or polyporous, measuring 3 times 10 to 15 millimeters. Using fine forceps, remove the minuting pin with the specimen from the mounting board and push it into the other end of the strip. Very delicate specimen can be mounted on minute pins set horizontally into a short strip of polyporous inside a gelatin capsule. Now this is a figure as showing the insect that's preserved with minuting pins. First, the minuting pin is pinned through the thorax of the insect, and then it is pinned on the entomological pin is pinned on the polyporous, and the insect along with the minuting pin is now pinned on the other end of the cork or the polyporous, and it can be preserved as long as possible with this method. The next method now is called gluing to pins. The insect is glued to a pin. Small ants and wasps can be glued to a standard insect pin or entomological pin. The right side of the thorax is attached to the pin with waterproof glue or nail varnish. So the insect is glued to a pin. It's as simple as that, and it can remain like this for as long as possible. It's a very simple but effective technique for the preservation of insects and arachnids, for the preservation of minute insects. The next method for the preservation of minute or small insects is called the gelatin capsules. The gelatin capsules are used for preservation of small hard-bodied insects. Small insects like parasitic wasps may be stored in gelatin capsules held on a standard insect pin. A small weight of cotton wool is placed inside the capsule to prevent specimen being shaken about. Smoothing and compacting the cotton wool block before inserting it helps to prevent the insects becoming entangled in the fibers. Smoothing and compacting the cottonwood plug before inserting it helps to prevent the insects becoming entangled in the fibers. Then this is the figure that explains the procedure or the insect that's placed on a gelatin capsule. It's also an effective way of preserving minute insects that are very small. The next method for the preservation of minute insects is called slide mounting. Small specimens have to be mounted on microscope slides so that they can be studied under a compound microscope. These include groups such as thrips, aphids, parasitic wasps, scale insects, book lice, and mites. All these ones are mounted on slides. Insects and spider body parts, example mouth parts and genitalia and liver, often have to be slide mounted and mounted on a slide for viewing. Microscope slide mounts may be temporary or permanent, but specimens maintained in collection require permanent mount. Each particular arthropod group requires a specialized preparation and mounting technique. So insects can also be preserved in microscope slides. Thanks for watching. Please continue to subscribe to our YouTube channel so that I can view very educative programs that will aid your understanding of biological techniques. Thank you.